All right, everyone. Welcome to the webinar, Putting Life Back into the Soil. So again, I'm Dave Stark. I'm co-founder and the head of agriculture for Holganix. And we're going to talk about the soil health journey. So this is one thing that we like to talk about, that bringing life back to your soil is something that I think, first of all, you're going to be amazed at what you see. It's not a sprint, it is a marathon. However, unlike a lot of things guys try to push on you, it doesn't have to cost you every step of the way. This is something where even year one, you're going to see some results and the profitability of the program. So again, it's the soil health journey. So this really is going to summarize what we do when we go out and work with customers and show them how putting life in the soil brings back function. So this particular slide, our product line is Bio 800 Plus. It has actually several thousand species of bacteria, actinomycetes, archaea, fungi, algae, protozoa, amoeba, even beneficial nematodes. Everything you see here is what the microbes in the soil do. Every picture here actually comes from a customer that sent the picture to us as a result using the product. So we're going to talk about all this. So when I talk about microbes, and I'm not going to go, oh, sorry, I'm not going to go into the depth of science I do in some of my other webinars. I know you're welcome, but I have to touch on a little bit. So these are some of the microbe types found in soil. And when we say microbes, of course, they're microscopic. You need a microscope to see them. If we look at the number in a gram of soil, and a gram is about the size of a chocolate M&M or a Skittles, if you like, the Starburst type candy. So you get the idea. It's not very big. And you can see the massive range of bacteria, which are the smallest, the fastest to grow, and like actinomycetes, which are bacteria-like, always hungry. They're the teenage boys of the soil. And then you get to the things that are larger and not as plentiful, fungi, protozoa, nematodes, etc. God put them all there for a reason. But now look to the far right column, the biomass in the top 6 inches of soil in pounds per acre. Look at the huge range. 350 pounds of bacteria up to over 2 tons. Fungi, we're going to talk a lot about fungi, almost nothing to approaching 7 tons. So this is kind of the difference between life in soil and not. And we're going to see, what that means. So before I get into it, one of the key recommendations we make, and probably the hardest conversation we have, is getting guys to back off their N, P, and K, to back off their fertilizer. It's really important because when a plant senses that it has enough nitrogen and or phosphorus, it stops feeding the microbes in the soil. This is how we actually start to degrade soil. We take it from the lively soil that has a lot of the microbes to now they have to fend for themselves. They eat the organic matter, the food in the soil, which actually degrades it. That's how organic matter goes down and down. We lose soil structure, et cetera, et cetera. How much can you cut your fertilizer? There's no set formula. I'm going to show you some examples, but remember, most people I work with are applying way above removal rates. So in a bushel of corn, there's about half a pound of nitrogen. Most guys I see, and they've been doing this for years, they're local agronomists that comes from the land grant universities, tell them put on a pound or more. We're applying too much. Same with P and K. So we can back down. So what does this look like? I like pictures, and you're going to see a lot of pictures in this particular presentation. This is a visual image, vegetative index of a farm in South Dakota. And you can see in the upper left-hand corner, that's just the satellite view. The lower right corner where it's kind of blue purple, that's his house. So it's not going to turn up green, his house and his yard and such. And of course, when you look at vegetative index, the darker green, the healthier and more robust the crop is. Yellow, less so, and red is bad. He used Holganix the first time in 2019. Look at the difference from in just 1 year from 2018. 2020 was a good year. Remember, this is South Dakota. He had a lot of moisture that year, so he had a really good crop. 2021, the opposite, was dry. Um, we need to get 2023 and 2024. In 23, he won the dryland soybean yield for the state, I believe Group Twos. What he did over this process, he started using over a pound of nitrogen per target bushel of corn he wanted to harvest. Now he's just over half a pound, and he applies no phosphorus or potassium. Now he has decent soil, especially today, and has a history of spreading cattle manure. There's a lot in the bank, so he has a lot that he could tap into. Can you achieve this? I hope so. But if your ground is lighter, sandier, maybe you can't cut this steep. Think of the product as a tool that helps you reduce fertilizer. And I want to give you more confidence in that. So this isn't just my science, and you're going to see just in the few science slides I have here, and in every deck I do, the fine print are the scientific references. So this is not something I made up. This is something that I look at the peer-reviewed published literature. So this slide summarizes 230 studies that look at how nitrogen is used in corn, rice, and small grains. I'll give you the punchline. It's right there in the middle in red. Biologically healthy soils produce greater corn yields per unit of fertilizer. Why? Well, when we don't have biologically healthy soils, the situation is like they're in the middle, surrounded by the black. We put nitrogen on the ground, the crop takes some of it up, but a lot of it sits there. It can volatilize away, it can erode away. Already, nutrient use efficiency, generally speaking, less than half of what we apply goes to the crop. It's worse for phosphorus, which is quickly bound up. But when we have biologically active soils, the situation is different. That's the picture there in the top right corner, that what happens is microbes are consuming the fertilizer and we get better efficiency. Let's take just an even closer look at this. So we start in the left-hand corner with fertilizer. It's usually some form of ammonia or nitrate, I mean, urea, etc. Remember the bacteria in actinomycetes. So in that kind of green picture, that's a picture of bacteria. They come in different shapes and sizes. That's a close-up of actinomycetes in that kind of reddish orange. They're the teenage boys. They're hungry. You put food out, they eat it first. And that's a good thing because they live in the root zone. So they immobilize your nitrogen right where you want it. It's a bad thing because of this thing called carbon to nitrogen ratios. For every nitrogen atom inside one of these guys, there's 5 carbons. To a biologist like me, that says it is an extremely high nitrogen containing organism. It's a pig. It ties it up. It immobilizes it, but it doesn't let go. So now let's look at the other things that should be in the soil. The upper right corner, fungi. And this is what I mean, filamentous fungi. If you have mulch around the house and you turn it over and you see all that white, that's beautiful. That's filamentous fungi. They are the best degraders of organic matter, your crop residue on the planet. They also eat bacteria. Their nitrogen requirement is 3 times lower than the bacteria they eat. Now we go to the bottom middle, the protists, the protozoa and amoeba. These are predators. They eat everything. One can eat 10,000 bacteria a day, but their carbon to nitrogen ratio is 30 to 1, 6 times lower. So they're eating a protein-rich diet, and protein is the main source of nitrogen in a living system, and it's toxic to them. So they have to spit it out. They spit it out as ammonia and nitrate. Where does this happen? Well, where does the food live? The bacteria in the root zone. What form of nitrogen does your plant take up the most readily? Ammonia and nitrate. I mean, this is God's design. The intricate dance here is unbelievable. And believe it or not, this is actually greatly simplified. Um, but it's not the end. There's more of what we have going on underneath us. I want you to remember this phrase: plants farm microbes, microbes mine the soil. In a healthy relationship, plants are secreting 30% or more of all of their energy to attract and repel specific microbes. They're farming them. What happens when we have enough microbes in the soil, not just the total population, but different kinds? They're always sending out biochemical signals. As we trigger a switch, it's called quorum sensing. And what happens is a lot of things immediately change in the soil. One thing is the tip of your roots start absorbing microbes. So your plants are actually eating the microbes in the soil. Now, when they do that, right at the base of the root tip, they secrete some things that strip off that what's called the cell wall, the outer covering around the microbes. They're protein rich, so they get a big dose of nitrogen. Now, that cell wall is there for a reason. One reason is to keep the cell from just bursting and dying. Well, a lot of them burst and die. Actually, most of them, when that happens, remember the nutrients that the microbes need, N, P, K, S, magnesium, iron, et cetera, all the micros are the same ones your plant needs. So when the plant absorbs a microbe and it bursts, the plant is getting a nutrient-rich diet. However, some of the microbes don't burst. Some survive. They move up the root to the bud of where the root hairs are. That's what those red arrows are. The microbes secrete a hormone that induces the root hair to elongate. They go through the root hair, back out in the soil, remine the soil, and the cycle goes again. Some of the microbes were even thought to go up to the above ground parts of the plant to cover the leaves and the stems. This is an amazing bit of work. It first came out of Rutgers University by Dr. Jim White. It's just stunning, the intricate complexity. And, this is where, if you've heard me before, as a molecular biologist, I personally, I've always had Jesus as my savior. I just thought he used evolution to make us. And then one day he taught me to actually use the brain he gave me. And I looked deep at the data and I was just stunned at what a fool I was. And I could go on and on. But, for example, the mathematical odds of a small protein, 150 amino acids, self-assembling. And if I cheat the math because it makes it easier for me, it's an odd of once out of a number followed by 191 zeros. 191 zeros to make one protein. That's not even life. You need 486 different kinds of proteins and more than one each to make life. All of the atoms in the known universe is a number with 80 zeros. That's a big number. But one out of 191, it's called absurdly impossible. This is God's design. And he made it easy for us to see. The evidence for him is all around us. All you need is a shovel. Or actually, if you're going to dig roots, we found a potato fork works better. This is a picture I got from a farmer in Iowa. What he was showing me is the plants, how much taller and greener they were, where he used our product, how much better the rooting was and the root hairs. That's what you see on the left three roots at the top. What I see is look at the other roots where we have only a nitrogen fixing bacteria added or starter fertilizing. They're white and they're bare. And if you zoom in, there's almost no root hairs. But you look at the ones treated with our product, there's dirt stuck to them and they're covered in root hairs. That says that there's not biological diversity in this soil until we add our product, which in and of itself, because of how much is in it, is a quorum. It triggers root hair formation, it triggers rhizophagy, and it triggers that dirt being glued to the root. And that's the action zone in the soil. And you could tell all this for the price of a little bit of your time. You look down at the soybean picture in the bottom. Now, they don't produce the rhizosheath, the dirt glued to the root like a grass will, but they do have nodules. And for years, people were telling me, man, nodulation is so much better with your product. And I thought, wow, well, I'm just dumb lucky. Nah, it's actually to form the nodules for the bacteria to be absorbed, and you probably use that inoculum. A lot of bacteria are absorbed, not just the rhizobia, and you need a quorum. Our product triggers the quorum, we get better nodule formation. So again, something you can see. Again, I gave you the nitrogen example. We can do this for P, K, etc. Your ground probably has years and years and years in there because we've been applying it and it's staying there. The plant root can't access it. Microbes can. It's one of the big reasons why plants farm microbes because microbes mine the soil. Now, guys will ask, so once I put life back in my soil, do I need to keep reapplying? And the answer is yes. You may through time be able to cut the rate. But there's a couple reasons. First of all, winter. Freezing kills the microbes because they're full of water. The water expands and it kills them, not evenly though. So we're way out of balance coming out of spring. By the way, I have some data I'm going to show you of what happens when you use our product and then stop in terms of the biological life and the soil structure. So we actually have that now. Tilling, we're going to talk about. Tilling is one of the worst things we do to the microbial life in the soil. Standing water, being waterlogged, deprives the ground of oxygen. That kills most of the microbes, and you can tell because of the smell. If it smells like rotting swamp water, that ground has died and it needs life added back. And of course, like I said, over application of fertilizers, fungicides, fumigants, but it's even more complex. Plants farm microbes. That means corn farms a little bit different microbes than soy, 5 to 10% difference. But it's more than that. Corn hybrid A versus corn hybrid B, 5 to 10% difference. Soy variety A versus variety B, 5 to 10% difference. Again, an intricate, elegant dance going on beneath our feet. Our product, again, has several thousand species of all the different types of microbes I spoke about. So it immediately adds a quorum and brings diversity back to the soil. You can tell if one of the products you're trying to be sold is diverse by the form. Our product, after we brew it, has to be refrigerated. We quick chill it, we keep it cold. And you can see the picture on the right, we shrink wrap it with an insulated wrap. You as a farmer don't need refrigeration. You got 6 to 8 weeks easy to work through it. We're not going to ship it to you in November to use it in April. We're going to ship it to you right before you need it. And if it's sat out too long, it tells you. Remember the waterlogged ground? You smell it? Well, our product smells sweet. There's a pound of molasses in every gallon. Our founder says it's a cross between Sam Adams and glazed donuts, and he's right, maybe with a touch of vinegar. If it smells like something died and is half decayed and you can't get your nose next to it, sat out too long, went anaerobic, the oxygen is gone, you just call us, we replace it. No questions asked. I don't care if it's your fault, doesn't matter. You're never financially at risk for product that's sat out too long. And by the way, we manufacture in the United States, outside of Philadelphia, Southeast Iowa, around West Point, and Kansas City. So we are American made. Now, do you believe everything I just said? I hope so, because I'm telling you the truth, but we put our money with where our mouth is, and that's what I'm going to spend the rest of the time talking about, is what we call our soil health tour. So we go to a customer's farm and we use a couple different tools. We use in the lower left-hand corner a probe from Crystal Labs. It's kind of like doing a Haney test real time that uploads to your phone in 30 seconds. Now, we're not doing so many of these that you can actually replace sending soil off with your agronomist to do Haney tests. We're trying to show you when you put life back in your soil, you're making changes. We do a microbiometer test that shows overall microbial life and the fungal to bacteria ratios. When we talk about tilling, it's the explanation why most farmed ground is heavily bacteria dominant. Remember the problem with bacteria. They're nitrogen pigs. They don't cycle the nutrients out. We need their predators, the fungi, protozoa, and amoeba, to keep cycling nutrients. We need them for other reasons too. We also use a leaf tech analyzer. Some of the things we're looking at, soil structure. We're going to show you changes in bulk density. Yeah, even after one application. Organic matter. pH, nutrient release, roots. We love digging roots. Remember, more roots, more water and nutrients explored because they're throughout the soil. By the way, we do all this. No customer gets an invoice. We want to come out and show you. You're on the soil health tour, on the journey with us. We're making changes to your soil, changes you're standing right there next to us that you can see. So this journey starts at planting in the spring. So our first product, what we call Bio 800 Plus Agriculture, we want you to apply it as close to planting as practical. You can do it with a burn down step pre-plant, with the planter, in furrow, 2x2, broadcast. I can find no statistical difference, even though intellectually these are soil microbes, and you might think in furrow or 2x2 is best. As long as you use enough total liquid carrier, like 15 gallons an acre, broadcast works fine. No later than V3. So if you have a choice, if you don't do liquid with your planter, I would rather you go early than go late. Now, if you're applying, a day or two after you plant, that's okay. But the longer the crop goes, and I'm going to show you why in these pictures, you lose some of the benefit. Remember, really, really, really reduce your fertilizer, take palatable bites. Don't go whole hog, maybe 10% nitrogen, maybe 20% P and K. It really depends where you are versus removal rates. This is a tool that helps you dial in your operation on your soils the right amount. But almost everybody can go a lot lower. We're also going to look at germination, stand, roots, stalk and stem strength, organic matter, cation exchange capacity, et cetera, et cetera. So, now just some pictures. These are all from us going out, taking pictures with customers standing next to us, or customers just sending pictures in because they're excited about what they see. This is Western Missouri. End of March, you can see the difference in germination between the treated on the left and the untreated on the right. Why is this happening? Well, it's because there are hundreds of species of microbes in our mix that are called plant growth promoting rhizobacteria. Fungi do this too. Remember, microbes live in the root zone. It's their home. It's where they're fed and protected. Without roots, they're cold, homeless, and hungry. So they secrete hormones that induce the seed to germinate into root. This is why I don't want you to apply too late. I'll show you in the next picture even more. Again, Western Missouri, 1 week later, look at, even though it's cold and it's frosting, look at the emergence and the root development. But look at this. This is Northwest Tennessee, planted March 30th, cold, wet conditions afterwards. Look at the difference. This product was applied in furrow. Which crop would you rather have? You know the key to yield is getting the feet under your crop as soon as you can and getting it out of the ground. And here we're showing we're like a growth stage ahead already. So I want you to have the benefit of this early germination and root mass, which is why if you have to go before and you can't go with the planter, that's my preference. Don't go way later because you miss this. Iowa. You can see the quote from the person who sent the picture, how surprised the brace roots are, and look at all the root hairs, the dirt stuck to the root. We've got biological activity, and all we needed was a shovel. Western Missouri. Now, he's right. I think it's harder to see in the picture, but he said in person you can definitely see the difference in the color and in the growth between the Czech and the Holganix treated. And this is West Central Indiana. By the way, I'm showing different geographies. I'm going to bounce around because I know you're from different places and you want to see something that's more relevant to you. And hopefully, I will show a picture or two that's close to where you are. Again, we're back to Western Missouri. Man, look at the difference in the color, the vigor. Importantly, we typically see the stalks and the stems are stronger, they're bigger around. A few weeks ago, I saw a presentation where corn yield was correlated with stalk diameter. Now, the thought, this is just a hypothesis, is that the bigger diameter stalk means there's more vascular tissue for water and nutrients to flow up and down. But this is pretty common that we can be even a growth stage ahead. Now we're Ontario, Canada, someplace different. Um, you can see the farmer's quote, way ahead of what's around. And that's a beautiful looking crop. Staying in Ontario. So this is another use of our product. This particular farmer had corn summer of 2023, planted a cereal rye cover crop, and then did the kill step with Holganix plus Roundup and planted beans. And you can see his quotes here. He planted into 4 foot tall cereal rye with corn stalks. Look at the emergence. So the biology is helping with the germination and stand, but look at the cereal rye and where are the corn stalks? The biology is chewing it up. That's great food, especially for fungi, and we're returning those nutrients to the soil. I'll give you some data on that later. Now we're into Pennsylvania, and this came from a longtime customer. And his notes there. Look at the difference in brace roots on the check 6, on the treated 11. And it's those brace roots or nodal roots that really are going to take over and start feeding the plant and helping with the productivity. Now this is a great story. This is Maryland, and by the way, in full transparency, this is one of the biggest differences in root mass I have ever seen. I hope if you use our product, you see this, but don't call and yell at me if you don't, because this is a phenomenal result. It's interesting. This is a very, very good grower in Maryland, and skeptical, which I totally respect. I've been in ag and agribusiness, ag technology 42 years, believe it or not. I know all the stuff being thrown at you guys. You should be skeptical. So he went out, we did the soil health tour, we had the potato fork, he's walking along, and as we're in the check plots, boom, boom, boom. And then we crossed the line where he planted Holganix, where he used the Holganix when he planted, swish. The fork goes right in. Swish. He dug these. What's going on? One application. Well, we'd have more data now. This is from Iowa. So we're looking at across 44 farms, the averaging bulk density and water infiltration. Look at the bulk density drop over a 7-week period. Look at the increase versus check in water infiltration after 7 weeks. 53% in beans, 64% in corn. It's unbelievable. Why? Why can we believe this? It's because soil structure is built by fungi and roots. Compacted soil is a biology problem. When you don't have the biology, clearly, when you put heavy equipment over wet ground, it's going to compact because there's no structure in the soil. When we have structure, it resists the compaction. So, a really, I think, good analogy is imagine you've got 2 5-gallon buckets. All soil starts out as really, really small particles called micro aggregates. Finely ground sand, we're going to fill that 5-gallon bucket with, and we're going to pack it down tight. In the next bucket, we're going to put golf balls. We're going to try to pack them down tight, not much will happen. Now pour a gallon of water on top of each. What happens with the golf balls? Oh, it goes in, takes it all. What happens on the sand? Puddles on top. That's what we want. We want the fungi, which produce the glue that makes those tiny soil particles, the microaggregates, and glues them together. And then that white feathery stuff, the fungal hyphae, they surround the big soil particles like the skin on a golf ball with plant roots, and they stabilize it. And that's important because the glue that the fungi make is a really good food for our hungry teenage boy bacteria. So it's now physically protected and now isn't going to compact. And as we look at this data, we apply the product sometime in April when we planted. As the roots and the fungi start to grow, they're producing the glue. They're taking the little soil particles, making big soil particles. The bulk density is decreasing, water infiltration increases. One application, you can track it. Now, here's pictures. On the left, this picture does not do it justice, I know, because I took this picture. This is close to my house, and this is on a path where I will go out and jog. These were some of the largest dirt clods I've ever seen in my life. Look at the face. It's sheer. These things are solid. What do you think's going to happen when water hits it? Is it going to go in? No. Can you just pick that up and crumble it? No. When the sun hits it, it's going to be a field of bricks. Contrast it with the picture on the right. This is a customer who'd been using our product for a couple years, and he's showing off his earthworms. But look at the texture where the sun hits that dirt clod. We have a grower in South Dakota that calls that kind of soil chocolate cake. It's moist and it's crumbly. That's the kind of dirt clod you want. So to move on with our soil health tour, again, we do the Crystal Labs probe to look at nutrients, bulk density, soil pH, microbial activity. So now we're beyond planting and we're getting into the season. And this is a summary of a lot of tests we did in the Peoria, Illinois area. I've got in yellow the things I like to look at. Again, the decrease in bulk density. By the way, if you're not familiar with the Peoria, Illinois area, it's some of the best corn ground God created. So this is already starting from a really good spot. But look, you can see differences even with one application in organic matter, a little bit better improvement in pH. Microbes are going to tend to move the pH to neutral. They will not substitute for an intervention if you have a pH problem, but they will help move things in the right direction. Microbial biomass at the bottom, we're seeing nice increases, and importantly, moving more of the microbial population to fungi. Because again, you generally don't have a bacteria problem in farm ground, it's lack of fungi. So now if we look at just the microbiometer tests from the Peoria area, and everywhere, I mean, these are individual farms where we went out and did tests. Um, in the yellow is where the check won. And by the way, none of this check ground really is bad, with maybe a couple exceptions. So the percent wins here is massive, but it's interesting. When we go out to the field with you, we will use the Crystal Labs probe. We'll take soil samples. We do the microbiometer test for these results later. We dig roots out in the field and we'll bring them back with us to show people. Two of the losing ones, these are the roots. Even though we didn't win in terms of microbial biomass and fungi, we still won with better rooting and better stalks. Now, some of that data, do you need to use our product every year? What happens? Well, you will make your ground better. So again, this is Peoria, Illinois, good ground. The table on top, we're still with microbiometer. MBC is microbial biomass carbon. That's basically measuring the total microbial load. And then you compare it to fungi and bacteria. The top, 2 years with Holganix, the bottom with 3. You can see it continually improves. And this is what we see. You're going to notice changes year 1. You're going to notice even more year 2 and year 3 and year 4. So we do want you to keep using the product. This is, I mean, the year 3 numbers are phenomenal. Now it's a fungal dominant soil. Unbelievable, with high microbial loads. Southwest Minnesota. This is another test. This ground, all of it was treated 2021, 2022, 2023 with Holganix. Year 4, some test strips were done. So look, even first of all, the treated, again, 3 years treated, just not last summer. This is some of the best bulk density, almost brings a tear to my eye. This ground is open. Look at the organic matter in the checks and the control. But even in year 4, we're getting a boost, a little bit of improvement in bulk density, a little bit better pH, a little more organic matter, and at the bottom, more fungi, more total microbes. And this comes from Ontario, Canada. Now, this is only 1 year. So in 2023, everything was treated. 2024, only part. And again, you can see with Holganix only, Holganix with starter, we're doing better both in fungi and the total microbial carbon. Now we're getting close to harvest. What are we going to see now? we see the plants actually stay green longer. And that's what we want. We don't want these things to die prematurely. We want our crop to naturally desiccate so that we're feeding the grain fill the whole time. And so we stay green longer. The grain dries down okay, that's not a problem, but the plant is able to feed it longer. So what do we get? Notice the last thing I like to talk about is yield. We sell, I mean, we're in the soil health business. We want you to have more open soil where someone asks you, how much rain did you get? You tell them all of it. I've got guys, because the soil is more open, use 30% less diesel because the tractor doesn't have to work as hard going through the ground. Their rows in the fall when they're applying anhydrous close because the ground is workable. Now, I'm not a huge fan of anhydrous. It kills everything it touches, but once it gets wet, it's going to be okay. I know why you use it. I'm not a big fan because it's going to kill what it touches, but it's not going to sterilize your soil. Just the band where you apply it. More yield data, healthy soil should reward you with better yield. It's not the measure I want people to use, though, because did you get rain at the right time? So many other things impact yield. Soil health, we deliver. Fertilizer reduction, we deliver. Your ground actually is better when you're done. How many times have I heard, and I know guys you want to leave the ground better for the next generation. We haven't had a way to do that, though, until now. And you combine this with other things you can do. Reduce tilling, because when you till ground, you kill the fungi. You break open those golf ball macro aggregates. Now the hungry teenage boy microbacteria eat the glue. We collapse the macro aggregate. We turn the golf ball back into sand. All these things contribute, but you have to have the biology too. Oh, and if you grow giant pumpkins, we got a guy that does that too. Um, so now let's go to after harvest. And I'm almost done here. We have another product that helps you break down your residue. It's also really important. Why? Your crop residue has a lot of nutrients in it. So for every 40 bushels of corn, you produce a ton of stover. In every ton of stover, you've got 17 pounds of nitrogen, 4 of phosphorus, 34 of potassium. Potassium is really important for stalk and stem strength, and some sulfur, 3 pounds. So a 200 bushel an acre corn crop, you've got 5 tons. That's a lot of fertilizer you already spent money on. Not one speck ever will be available to a future crop unless a microbe breaks it down. So we need to have microbial activity. To have microbes work, we need some temperature and moisture. So when you're going to use a breakdown product for residue control, you don't want to use it once it's winter, the ground is frozen, it's not going to thaw. Again, a little bit of temperature and moisture helps. The other thing that's huge with products like this, remember the microbes that are most effective at breaking down residue are fungi, certain fungi, white rot, brown rot, black rot, not unicellular fungi like yeast. Bacteria don't do a good job. Why? 2 of the 3 most abundant polymers on Earth are called cellulose and lignin. It's like rebar and concrete. It's what makes a 100-foot tall tree stand up to that 90-mile-an-hour crosswind. It's really hard to degrade, but not for fungi. Let me show you some pictures. So our breakdown product is enriched for these rot fungi that are designed to break down your residue. These photos are 10 days after application. Our soil health tour, now we're back to Peoria, Illinois area. Again, you can see the corn was harvested early because he had a dry period and it had matured. And after basically 9 days, you can see already growing inside the corn stalks, all that white, brown, and black rot fungi. And remember, you won't always see it driving by on the road because the fungi are going to do the rot from the inside out and also within the mat where you can get morning dew and there's more moisture. So go out and pick it up. What we want is by the time you plant in the spring, you're just planting right through it. It just crumbles. So you don't turn the soil over. Don't kill the fungi. Don't ruin your soil structure. We want the fungi and the balance out there. So again, to plant right through the residue. And again, you'll plant faster. Everything's going to work better. Um, I've got guys literally that have sold their chisel plows. Will never chisel plow again. One of the best things they're going to do for their ground. And if you happen to plant a cover crop in the fall or winter wheat, that breakdown product, just like the spring product at the beginning of the journey at planting, it will help with germination and stand of your fall crops. So again, our two products, spring, Bio 800 Plus Agriculture. You know, when we brew our product, and I should give you just a little explanation, we start with a very specific starter that's all plant-based. And we change over 2 dozen variables in our manufacturing process. They all grow together, the thousands of species. So they're cooperative. They don't fight each other. Oh, they will use each other as food, but that's natural. We actually want that. But the thousands of species, we can tweak the final outcome. So in the spring, we are promoting those kinds of microbes that produce the hormones for germination and rooting and really help with fertilizer use efficiency. In the fall, we enrich for the white, brown, and black rot fungi. The biology overlaps tremendously between the two products. It's just what we enrich for. They're the same price. So next year, we're going to do this again. We did thousands and thousands of studies, soil samples this last year. It's really an exciting time that there's a tool that will help you produce more and use less while you actually leave the ground better for that next generation. So I want to thanks. I gave you a lot. If you've got questions, Ritu, I am happy to answer them.
Dave, our first question is, does our product contain ar archaea?
Archaea?
Yeah.
Archaea? Yeah. Um, Archaea are a kind of bacteria. I know a lot of them tend to be found in really severe environments, like volcanic vents and such. They're just interesting. They're within the broader classification of bacteria. And by the way, I should have added after every batch that we brew, and Ritu, of course, you know this, because you look at the tests, we send off a sample to be DNA fingerprinted. So we have a third party lab that helps us with quality control.
So another question is, what do you think of deep subsoiling to get oxygen into the lower soil levels and increase deep carbon sequestration start?
Yeah, I would rather do that unless you've got a lot of nutrient stratification or you've got, some compaction layers that are just a real problem. If you need to do a deep rip, then do it. But I would rather you do that with different kinds of crops and let the roots create the channels for the water and the air. And we have, I've got some customers that find fungi from our product 5 feet down. So it may not happen year one, but in time, that root mass is amazing for the soil because when those roots die, they're not only food for microbes, but they're air and water channels so that we aerate and wet the ground naturally.
What about fungicide being applied with the planter 2 by 2 or on the seed? Is that an issue?
No, it's not a seed. And you may have noticed one of my earlier slides, the corn seed had a seed treatment on it. And I'm not worried about fungicides on the seed coat. I don't want you to tank mix with fungicide. That's pretty counterproductive. And if you use a multi-mode of action fungicide like Xyway, just keep it apart. Put our product in the furrow and use the Xyway 2 by 2 or apply us broadcast. Just keep them separate.
Mhm. How close to killing a frost can you apply in the fall with?
Yeah, a light frost doesn't bother me because when you apply the breakdown product, again, we want it wet into the residue mat. So the temperature underneath that mat is still going to be okay. And sunlight, because I went out this fall and looked at some residue mats and even did a few short videos, sunlight's going to warm that residue mat up. So until you're really below freezing, I'd say 25 degrees and lower, you're still going to get some breakdown. Now, I lived 36 years in St. Louis. I would say most winters we had enough sunshine and warmth that we would see degradation all year. Now, this last year was a little bit cold. So maybe not this year, but it was unusual. So I'd say if you're north of north of I-80, then you probably don't want to go past Thanksgiving in a normal year. But just use your judgment. 2 to 3 weeks where we're 40 degrees with a little sunshine and moisture, you're going to see the fungi grow. And they're tougher under those conditions than bacteria. So it won't be as bad.
Have you analyzed the potential increase in photosynthesis with Bio 800 Plus?
I haven't. Now, back when I was actually a scientist, I did, working with my own hands, I did work on what's called source sink interactions, which, the source will be the leaves that are creating sugars from photosynthesis using atmospheric CO2, water, and sunshine. The sinks are the places in the plants, like a potato tuber, the flower parts, the growing parts that are demanding this. In almost all plant species, photosynthesis is not the limitation. It's demand for photosynthate. So I don't have the same feeling because of the work I did genetic engineering these pathways with my own hands that photosynthesis is what we focus on. We have to create demand for the products of photosynthesis. For example, in potato, you can mimic insect feeding and remove 80% of the leaves. Photosynthesis kicks way up in the remaining 20%. So it's more demand limiting, and that's the way biochemical pathways work. So products of biochemical reactions tend to slow down or stop the reaction because it's a signal you got enough, don't do anymore. If that product is removed, the biochemistry keeps chugging. There are a few exceptions. Some plants are photosynthetically limited, generally not grasses, though. Grasses are pretty efficient.
Where do we source slash buy your product? How much is it typically per an acre?
Okay, so we have sales reps and dealers across the country. So I know you can go to the website. You could certainly contact me. We manufacture in Pennsylvania, Iowa, and Kansas, the Kansas side of Kansas City. It's $40 a gallon, $20 an acre. And I would urge most people reduce your fertilizer by at least that. It's a good rule of thumb.
Is there a concern if we use litter 3 tons per an acre during spring applied?
No, the microbes love chicken litter or manures. I would just say my concern would be so for example, I've worked with guys a few years ago that would put down 300 pounds of nitrogen and spread anywhere from 10 to 15,000 gallons of hog manure. I don't even want to sell them my product because those plants will never feed the microbes. There's way too much fertility. So as long as you're balancing your total nutrient load with the chicken litter and dialing back on the synthetics, use it when you need it, especially in season. If your crop's hungry, feed it. The microbes love chicken litter. It's just a really good food source for them, and it will help break it down faster and release those nutrients. Well, thanks everyone. I really appreciate your attention. I know I gave you a lot. If you've got questions, um, I think you can find me on Holganix.com, and Ritu, I know you'll make available a recording.
Yep.
Okay. Thanks everyone.